So we did the receptor signaling molecules, we did the receptor classification, and we have done already tyrosine kinases. Now we are moving on to the final part of our signaling topic, that is the JAK-STAT pathway. Now, the JAK-STAT pathway is specifically opted by the cytokine receptors. Now, when we are talking about cytokine, remember, just like hormones are chemical messengers of the endocrine system, the cytokines are the chemical messengers of the immune system. So the cytokine receptors might include the cytokines. These receptors which follow the JAK-STAT pathway might involve the cytokine receptors. Some of the hormones, such as the growth hormone and the prolactin, also follows this Janus kinase pathway. Now, as you see that I have said that let us start, when I started, I said that let us start about the JAK-STAT. So these are two molecules you can understand. One is the JAK, which is Janus kinases. The name Janus is after the Roman god Janus, who has two faces. So when you look at the JAK molecule, it has two faces. On one hand, it can interact with the receptor, and on the other hand, it interacts and further activates the receptor. So it has two. So drawing a simulation with the Janus god, this name is given as JAK, which can phosphorylate and activate gene regulatory proteins, which are known as STAT. The full form of STAT is signal transducer and activators of transcription. As you can see from the picture, Janus kinases are part of the receptor, while STAT proteins are located in the cytosol and are referred to as latent gene regulatory proteins because they only migrate into the nucleus and regulate gene transcription after they are activated. So in the inactivated condition, they are present in the cytoplasm, and following activation after a signaling stimuli, the STAT proteins moves from the cytoplasm to the nucleus. Now, if we start with the JAK, these cytokine receptors are present in the plasma membrane as dimers or trimers, and they are stably associated with one or two of the four known Janus kinases, JAK1, JAK2, JAK3, and TYK2. Now let's see the mechanism. See, this is a cytokine receptor. And as you can see, the cytokine receptor is present as a dimer, but it is inactivated. Now, whenever the ligand comes and binds with the cytokine receptor, you can see that ligand binding causes conformational change in the cytoplasmic site of the receptor, as a result of which the Janus kinases domain, the kinase domain, if you remember, kinase means addition of phosphate. So the kinase domains are brought together, which then phosphorylate each other on a tyrosine residue in a region called activation loop. So we have this activation loop, which is a tyrosine kinase domain, where a tyrosine residue will be phosphorylated. They are present as dimers, you can see, but they are not activated. The moment ligand binds, there's a conformational change occurring here. As a result of this conformational change, the kinase domains of the JAKs are activated and they phosphorylate each other on the tyrosine residue in a region called the activation loop. Once they have become activated by phosphorylating the tyrosine in the activation loop, then this activated tyrosine residues further phosphorylate tyrosine residues on the receptor. So initially they are phosphorylated and activated, JAKs are phosphorylated and activated. And once they are phosphorylated on tyrosine residues and are activated, they in turn phosphorylate the tyrosine residues on the cytoplasmic end of the receptors. So as you can see, tyrosine residues are phosphorylated. The moment the tyrosine residues on the receptors are phosphorylated, as you can see, the cytokine receptor is now activated and it will now recruit the stats. Earlier, last class, we had studied the SH2 domain. So the SH2, the stats with their SH2 domain will come and interact with this phosphotyrosine molecules. Let's look into that picture. So you can see the cytokine receptors have been activated, and this is a STAT protein. If you look at the structural composition of the STAT protein, it has a blue color SH2 binding domain, a purple color DNA binding domain, and you can see the tyrosine residue that will be phosphorylated. So what happens? The STAT molecules interact via their SH2 domain with the activated phosphotyrosine residues on the receptor. Once they interact, they in turn are phosphorylated. You can see they are phosphorylated. So they are activated. The phosphorylated STATs then dissociates from the receptor. So they have come out from the receptor. And what happens? They dimerize. So the monomers, they initially are present, they are phosphorylated, and then the phosphorylated STATs come out of the receptor and they dimerize because the STAT homodimer has two phosphotyrosine domain SH2 domain interactions. So they dimerize where the STAT complex is stabilized. Once this stabilization has occurred, once the STATs has been phosphorylated and dimerized, they now do not associate with the receptor anymore. They move down into the nucleus. As they move down into the nucleus, you can see this is the DNA. They bind to the DNA. If you remember, there was a DNA binding domain. So the violet color, see the violet colors. So they bind to the DNA via their DNA binding domain. And as a result of binding to the DNA via the DNA binding domain, they bring about the activation or the transcription of the genes to whose response element they bind. This is another very simplified and easy picture. See the cytokine receptors. See the janus kinases associated with the cytoplasmic end of the cytokine receptors. And once the cytokine binds, you can see the receptors are activated and the janus kinases are phosphorylated in their activation loop. They in turn phosphorylate the tyrosine residues on the receptors. You can see that the STAT molecules then associate with the phosphorylated tyrosine residues via their SH2 domain. The STAT monomers are activated, they come out from the receptor and dimerize via their SH2 domain. They move into the nucleus and bind to the response area of the target gene, bringing about the activation of that particular gene. So this was a very simplified diagrammatic illustration of how once a cytokine, or as I said, some growth hormones, the hormone prolactin binds to the receptor, in the process activates the janus kinases associated with the receptor, which in turn activates the receptor itself by phosphorylating tyrosine residues on the receptor. The cytosolic protein STATs are then recruited to the receptor via their SH2 domain. The STATs are phosphorylated on tyrosine residues. The monomers open and dimerize via their SH2 domain. They are stabilized, they move to the nucleus and bring about the response of the target genes. This is another very simplified picture of the same signaling pathway. A cytokine receptor, genus kinases are phosphorylated, phosphorylating the tyrosine residues on the receptor. STAT molecules are recruited here, and STATs are phosphorylated on the tyrosine residues. The STATs then interact via their SH2 domains and dimerize, bringing about specific gene transcription. So this was about STAT. This is another simplified picture of STAT as STAT moves in and activates the target gene. The target gene is transcribed, translated to give the desired protein. And these are some of the signal proteins that act via the STAT receptor, the interferons, the erythropoietin, prolactin, growth hormone. And these are some of the physiological responses that are generated since the cytokines basically act through these receptors. So it plays a big role in immune system when interferons are the stimuli. When erythropoietin is the stimuli, production of erythrocytes. If prolactin is the stimuli, milk production. If growth hormone is the stimuli, if growth hormone is the stimuli, then we have stimulus growth by binding to IGF-1 production. And when we have GM-CSF at the stimulates the production of granulocytes and macrophages. So with this, we come to the end of JAK and STAT, and we come to the end of also a signaling topic.